Hello, everyone! Welcome to today's micro class. Today, we will continue our study of leaves, focusing on leaf arrangement, phyllotaxy, and modified leaves. Leaves are arranged on the stem in a regular pattern, which is known as phyllotaxy. There are four main types of phyllotaxy: alternate, opposite, whorled, and clustered. Alternate phyllotaxy is characterized by one leaf per node, with leaves spirally arranged along the stem. For example, the leaves of mulberry and peach trees grow alternately on the stem. Opposite phyllotaxy features two leaves per node, arranged in pairs. Syringa, lilac, and dianthus, carnation, have opposite leaves. In some cases, such as in mint and gentiana, gentian, the opposite leaves are arranged in a cross shape, known as decussate opposite. In other plants, like metasequoia, dawn redwood, and ligustrum, privet, the opposite leaves are arranged in two rows along the stem. Whorled phyllotaxy involves three or more leaves per node, arranged in a whorl. The leaves of Nerium oleander, and Stemona sessilifolia, Mick, are typical examples of whorled leaves. Clustered phyllotaxy occurs when two or more leaves are clustered on a very short internode. Ginkgo biloba, ginkgo, Lycium barbarum, goji, berry, and larix, larch, have clustered leaves. The stem that bears leaves and buds is called a branch. The part of the stem where leaves are attached is called a node, and the section between two adjacent nodes is called an internode. You can clearly see the nodes and internodes in the branches of Ilex cornuta, big leaf winterberry, and Ginkgo biloba. In some plants, the stem is extremely shortened, and leaves grow from the base of the stem, seemingly emerging from the roots. These leaves are called basal leaves. If basal leaves form a rosette, they are known as a rosette leaf cluster. The leaves of Taraxacum officinale, dandelion, and Plantago asiatica, plantain, are examples of basal leaves. Next, let's explore leaf modifications. Over the course of evolution, leaves have undergone changes in shape and function, resulting in various modified forms. Bracts are modified leaves that grow below flowers or inflorescences. The multiple layers of bracts surrounding an inflorescence are called involucral bracts. For example, the green involucral bracts of sunflowers and the spathe of Xanthodeschia ethiopica, calla lily, and alocasia, elephant ear, play important roles in flower development and protection. Scales are leaves that have become specialized or reduced to a scale-like form. There are fleshy scales, such as those in Lilium, lily, Fritillaria, fritillary, and Allium cepa, onion, which are thick and succulent. There are also membranous scales, like those in Ephedra, Ma Huang, Sagittaria, Arrowhead, Eleocharis dulcis, Berm, F. Trin, X. Henschel, and Allium cepa, which are thin and translucent. Spines are leaves or stipules that have become modified into sharp, needle-like structures. The spines of Berberis, barberry, and Opuntia, prickly pear cactus, are examples of leaf spines, which protect the plant from external damage. Tendrils are leaves or stipules that have become modified into slender, coiling structures. The stipule tendrils of Smilax, Sarsaparilla, and the leaf tendrils of Pisum sativum, P. help the plant climb and attach to other objects. In contrast, the tendrils of Loupha, and Trichosanths, snake gourd, are derived from stems and should be distinguished from leaf tendrils. Phyllodes are petiolets that have become modified to resemble leaf blades and take on the functions of leaves. The phyllodes of Acacia confua, Formosan koa, are an example of this modification. Insectivorous leaves are specialized leaves of carnivorous plants that capture insects. Plants like Nepenthes, pitcher plant, Dionaea muscipula, Venus flytrap, Drosera, sundew, and Sarracenia, trumpet pitcher plant, have unique insectivorous leaf structures. Finally, let's take a look at the internal structure of leaves. The structure of dicotyledonous leaves includes the epidermis, mesophyll, and veins. The epidermis has dorsal and ventral sides with stomata present, especially more on the lower epidermis. The mesophyll is divided into palisade and spongy tissues. The veins, including the midrib and major lateral veins, consist of vascular bundles and mechanical tissues, with xylem located above and phloem below. For example, mentha, mint, has dorsiventral leaves, while the structures of Nerium oleander and Cassia angustifolia, senna, have their own unique characteristics. The structure of monocotyledonous leaves, taking Poaceae as an example, features more regular epidermal cells, including long cells and short cells, as well as bulliform cells. The mesophyll does not have a distinction between palisade and spongy tissues and is isobilateral. The veins are composed of limited exarch vascular bundles and are generally arranged in parallel. You can clearly understand this by looking at the cross-sections of Oryza sativa, rice, and Z. maize, corn, leaves. Today, we have learned about the types of leaf arrangement, leaf modifications, and the internal structure of leaves. This knowledge helps us better understand how plants grow and adapt to their environment. I hope that after class, you can observe plants more closely to reinforce what you have learned. Thank you for watching, and see you next time.